If we now begin with, they term it carbohydrate metabolism, but really, what do we do with glucose? All right. And so what you know to do with glucose at this point in time is degrade glucose in a manner to produce as much ATP as possible. All right. And so we take glucose and make pyruvate. That pathway that takes glucose and makes pyruvate is what pathway? Glycolysis. Okay, and that's the one pathway that we learn most often. This particular chapter is going to take glucose and we're going to do a couple things to it. We're going to degrade glucose, but in a totally different way. Okay? Yes. I will at the end. I need to get to a certain amount, and then I'm going to leave some time for it. Okay. We're going to degrade glucose in a different way than we did before. Okay. We're going to take glucose, which is a 6-carboned sugar, and we're going to make a 5-carbon sugar called ribose. Okay. As we do that, we're going to make NADPH, which is going to be used to reduce items in synthetic processes within the body. Okay? Ribose is important because why? What? You need it for RNA, and ribose can be modified to make DNA, and ribose is a component of the electron carriers, FAD, NAD, and ribose is a component of CoA, and ribose has a lot of place. It's very important. A lot of very fundamental structures have ribose as its core. Okay, so making that's important. We're going to do that using something called the pentose phosphate pathway. Have you heard of the pentose phosphate pathway? No? You're going to know it after today. Okay. Now, we're also going to look at the synthesis of glucose from citric acid cycle intermediates and from fat precursors, although that's kind of more limited than you think. All right. And we're going to see the synthesis of glucose from lactate, because remember, lactate was a dead-end product. It was the anaerobic anaerobic product of glucose, and it's a dead-end product, but we'll see how we can resynthesize glucose back. And then we're going to talk about glucose as storage in the form of glycogen. And glycogen was a polymer that we looked at last chapter, and it's a storage molecule in human. We have similar starches that are similar in structure, but we're going to talk about the synthesis and the degradation, and we're going to talk about the regulatory events that control that, because glycogen is going to keep the blood glucose levels constant so that your brain can function. And so this is very highly regulated about how it's used. All right, the pentose phosphate pathway. Okay, lovely little thing, the pentose phosphate pathway we're going to deal with. Okay, obviously we're going to make pentose. It's got a phosphate on it, and oh my goodness, this is going to be a pathway. All right, and it's going to be one pathway in which you're going to have to decide which way this pathway goes based upon what the body needs. It is not one singular pathway. It is multiple different ways that we can go, and it's going to kind of be a circle, and depending upon what the body needs, I can go back and forth and up and down through these pathways. Okay, I will teach it the normal presentation, and then I will give you some situations in which it might work differently. All right. It ultimately has the ability to generate NADPH, which is a reducing equivalent, in that it donates hydrides. Okay. Up to this point, we've had electron carriers that accepted hydrides. That's in the oxidation of our food. Okay. We're also going to generate, as the outcome that is possible, ribose 5-phosphate, which is a precursor to a lot of different very fundamental structures. Although I do not have to generate either one of those, okay, or I can generate both of those using the steps in the pentose phosphate pathway. Okay, there are two main steps in this that are identified. One is the oxidative portion. These are the reactions that produce the hydride containing molecules, the NADPH, and it will end up with a sugar called ribulose 5-phosphate. Then there are the non-oxidative reactions. We're going to produce the ribose 5-phosphate, which is what I told you it produces, and then it's going to manipulate what is produced and produce glyceraldehyde 3-phosphate and fructose 6-phosphate, which are intermediates of two processes, one, glycolysis, or two, gluconeogenesis, of which we're going to look at gluconeogenesis after the pentose phosphate pathway in detail. All right, let's take a look at the pentose phosphate pathway and its first reactions. Okay, we started with glucose 6-phosphate. We know that, we love it. We could draw it if we had to, right? Okay, my G and my 6 always looks the same. Okay, and so this enzyme is glucose 6-phosphate dehydrogenase. Okay, its number is 1.1.1.49. Okay, and you can look that up. There is a base that is at work, and we're going to have a hydride move. That's what dehydrogenases do. They have hydrides. Now, in this case, the acceptor is going to be NAD+. And so I'm going to have a base. Now, the actual base in this is a histidine. Okay, but I'm not going to draw histidine because that's way too much drawing. I'm going to just say base. Of the two, if you then remember, look at your reactions, you got this and that. You can see right here doesn't look like this, does it? I've got an oxygen bound to a carbon, I've got an oxygen bound to a carbon, but here this oxygen is missing something, right? And this carbon is missing something. And so the base is going to be able to take one of them. One is more acidic, and that would be the one that is bound to the oxygen. The reason is, if I take the proton off, the oxygen becomes negative for just a second. If I took the other one off, what becomes negative? The carbon. In terms of carbon wanting an oxygen and oxygen wanting an instead of carbon wanting an electron and oxygen wanting an electron, the oxygen is much more electronegative, and so it is easier for the reaction to proceed right there. This pair of electrons, even though it can be temporarily completely on the oxygen, it will flow to form the double bond that is produced. The hydride that leaves is the proton at the top to produce NADPH. Okay? I get a lactone, and the next enzyme is gluconolactone, and it's going to react water to this and open up the ring. Now, again, a histidine will act as a base. All right, and it's going to first react with water. Now, oh my goodness gracious, if you were trying to figure out what in the world this here becomes right there, okay, I have no idea what's going on, right? Because it's the first time I've ever seen this. So, what you do is you start numbering things. I can identify that one because that's the phosphate on it, okay? And so, the next carbon would be 2. The next carbon would be 2. The next carbon would be 3. There's 3. Here is 4. There is 4. Here is 5. There is 5. And this one is 6. And that one is 6. All right. So, what's going on here? 5 and 6 carbons. And then I got this thing, which would be number 7, the oxygen. Well, where in the world is that? Okay. This is what we're going to see where that oxygen is actually going to reside. The oxygen 7 is actually bound to carbon 2. So the oxygen 7 is that. Okay? So that's kind of interesting. All right. We're going to really physically break, all right, this bond right here. Because the 7 oxygen was bound to the 6 carbon, and that's not what happens when it gets at the end. So that's where the breakage is going to happen. The base will take the proton off of the water to create a hydroxide. The hydroxide will attack at the carbon of a ketone. Why would a negatively charged hydroxide attack a ketone? It's partially what? Positive. All right. Now, every time I attack a ketone, I can do a couple things. I have to kick up, all right? And at that point in time, I have 2 alternatives that I know of right now. I can kick down, or I can pick up a proton. Okay? What would you think this does? It kicks back down because in the product I still have a ketone. So I have to reform that ketone, and this picks up a proton. All right, that proton that I just colored in blue is that one. Okay. The oxygen from the water is this oxygen here. Uh-oh. What did I write, and what, in terms of the mechanism, and what did I write at the product? What is missing? I attacked with a hydroxide, right? So up here, I should have written double bond O, oxygen, and a proton, right? Because that's what I wrote in my mechanism. But why is the proton missing? What would this functional group be? An acid. Acid pKa's are around 4. What is the pH of the solution in the cytosol? 7.4. And 7.4 is so basic compared to that pKa, it's going to deprotonate it. And so you very rarely ever find acids in the protonated form in the body because of a pKa argument. All right, I opened up the ring. The next is a 6-phosphogluconate dehydrogenase, and again I have an NADPH. And so inventorying, same at the top, same right here. Okay, hydroxide here, no hydroxide there. There's where my reaction is going to be because all of this is the same. So it's around this bond. Okay, a base will take off that proton. Electrons form the double bond, and a hydride is accepted by the NADPH. There is an intermediate structure because I am going to get to ribulose. How many carbons are in ribulose? 5. And how many carbons did I start off with? 6. So what falls off? Carbon dioxide. Okay. A carbon dioxide, remember, is C double bond O with 2 pairs of electrons on both ends. Okay. Well, that looks like that and has 2 lone pairs. But this one has 3 lone pairs in that negative charge. It almost looks like CO2, right? Well, let's make it look more like CO2. Put that there. Okay. Now, what we have in this reaction is called a oxidative decarboxylation. Okay. The oxidation happens first. The decarboxylation happens second. Why in the world? Okay. Let me go back for just a second. What if I did this? And I said, that went off, and then I put a negative charge on that carbon. Negative charge on a carbon is not good, we said, right? So that doesn't happen, okay? But I just did this, and now this pair of electrons that is right here could go onto that carbon. But that we said that was bad, right? But because I have this double bond ketone here, what could happen is a resonance structure. Okay? Where now the negative charge is sitting on an oxygen, not a carbon. All right, so what I get here is a whole bunch of mess. Okay? I get a double bond that has a hydrogen, that has a hydroxide, that has a carbon, and an oxygen with a negative charge. Then I have all that, the R group that's not as part of the reaction. All right, now let's take a look. This is this carbon right here, do you see? And then this carbon is this one right here. It needs to be a ketone. That's not a ketone. This carbon needs to have a hydroxide. It does have a hydroxide. It needs to have a hydrogen. It has a hydrogen, but it needs to have one more. And so we always have a proton in the mix. And now what happens is the electrons will reform the ketone, and this double bond can pick up the proton, and we're at our product. The fact that I created that ketone allows the decarboxylation to happen because I get an intermediate resonance structure that stabilizes it. If that wasn't there, the acid doesn't fall off. Okay. I have ribulose 5-phosphate. This is the end of the oxidative steps, although some textbooks put this particular reaction as part of the oxidative steps, but there's no oxidation going on. Okay? This is an isomerase. Remember what isomerase mechanisms are? Well, if you look at ribulose 5-phosphate and ribose 5-phosphate, and you count out the number of carbons, the number of oxygens, the number of phosphates, the number of hydrogens, they're all exactly the same. Okay, we're just isomers. So the mechanism says, take off a proton, add a proton, take off a proton, add a proton until you get what you want. Okay, now there is a stability issue, and the reason why we actually end up between the two. Oh, I do want to correct something. These are in equilibrium because you can go back and forth. All right, if I take a look at what's going on, I can see the carbon up here that is number 1 has 2 hydrogens, and the intermediate, it doesn't have 2 hydrogens. So let's take it. And I form the double bond, and I form a hydroxide, okay, because every time I kick up onto an oxygen, I could either kick back down or I could pick up a proton. Why did I pick up a proton here? Because a proton is found in the intermediate. Now, this surely doesn't look like that, but we can make it look like that. Okay, I need to get rid of a proton because the oxygen is not supposed to have it. This pair of electrons forms the double bond, but I have too many bonds to a carbon. That carbon picks up a proton, and so this is the proton I just picked up. Okay, and the ketone is made, and I have an isomer, ribose 5-phosphate versus ribulose 5-phosphate. All right. I told you we produced two things in the pentose phosphate pathway. What are those two things? Ribose 5-phosphate, I said, this is what we make, plus NADPH. But how many did we make from glucose? 2. We made 2 NADPHs. All right. Now, if you think back to NADH, how many ATPs can you make from an NADH? 2.5. Okay. The hydrides that are coming off of this have the same equivalent amount of energy. So we extracted quite a bit of energy out of these two reactions. Now, ribose 5-phosphate can be used to create DNA. Can it be used in this form to create DNA? Do you see ribose like this? No, it can cyclize. Okay, and most of the ribose is found in a cyclized form. All right. So we can say that from glucose 6-phosphate we get we will create 2 NADPHs, 1 ribose 5-phosphate, plus a CO2 and a proton. Now, every time we made energy from glucose in glycolysis and the citric acid cycle, the waste product was CO2. And so we have produced some waste but extracted some energy. Okay? We do not have as many carbons as we started with. So the potential energy has been extracted from it. All right? And so we have done what is the classic pentose phosphate pathway. We've made our two things. Now, there's a non-oxidative pathway, okay, in which sometimes I don't need the ribose. Okay? If I don't need the ribose, then I take 1, 2, 3 equivalents of ribose, and I do a couple reactions with them. And what I will do is I will produce a fructose 6-phosphate. I will produce a fructose 6-phosphate, and I will produce a glyceraldehyde 3-phosphate. Okay, let's do some higher math. 5 plus 5 plus 5 is what? 15 carbons come in as ribose equivalents. Okay? I didn't say ribose, I said ribose equivalents. All right? Fructose 6-phosphate has how many carbons? 6, and another fructose 6-phosphate has another 6. So that's 12, plus glyceraldehyde 3-phosphate brings in another, how many? 3. 12 plus 3 is 15 carbons. Okay? The enzymes that are used are transaldolase and transketolase. How many carbons are transferred when transketolase does its reaction? 2. Okay? I will take 2 from this one and add it to here. I get 7 and 3. Start with 4 and I start with 5. I will take 2 from this one and put it onto that one and get 6, and that will be left as 3. How many carbons are transferred when I use transaldolase? Be careful. How many carbons are transferred? 1 is a possibility. What's the other possibility? Yes. Okay, it could be 1 and it could be 3. All right. It is actually 3 that is moved. And so I will take 3 from here and add it to that. That becomes a 6. This becomes a 4. Okay. All right. That's a biology textbook of looking at it. We're going to look at it a little bit closer. Okay. The transketolase transfers a 2-carbon unit. The transketolase will transfer a 3-carbon unit. Okay. The structures that happen, okay, are ribose equivalents. Okay. Now, we said ribulose could be transformed into ribose, right? By an isomerase. Okay. Ribulose can also be transformed into something called xylulose using this enzyme is an epimerase. What is an epimerase? It changes the sugar at one hydroxide's position and switches it, because those are related as epimers, and so an epimerase just switches those. Okay, so here is the big deal, and this is something that you need to know. The ribose 5-phosphate is an equivalent to the ribulose 5-phosphate, which is an equivalent to the xylulose 5-phosphate. They are all isomers of each other. They are all 5-carbon intermediates. But since it is the pentose phosphate pathway, any one of those three, since they can be interconverted, a lot of times a book will call it a ribose equivalent. Okay? And so they are 3 separate molecules, but they are the 5-carbon intermediates that can be interchanged within the body. So this is an important idea. And so, to go down the non-oxidative pathway, we do use one ribose 5-phosphate, and then we use the ribose equivalent of xylulose. It's because the enzyme has to have the right orientation of the hydroxides to do the reaction. It will take 3 carbon, the transketolase will take 2 carbons, okay, and it takes, which of the two? These two. It takes these two, plucks them off, and puts them right there. So you go glyceraldehyde 3-phosphate and ceduhepulose 7-phosphate. Isn't that just wonderful? Okay. Oops, did I go too far? No. Stop. All right. Then I re-react the glyceraldehyde with a ceduhepulose using transaldolase, and I take the 2 that I transferred plus 1 other one, and I put it back onto this one to make fructose 6-phosphate and something called erythrose 4-phosphate. Now, this is good because I know what to do with it. Okay? I'm in either glycolysis to break down the sugar, or I'm in gluconeogenesis to make sugar, to make the glucose. Now I got this 4-carbon thing that I have no idea what to do with. I bring in another xylulose 5-phosphate, which is a ribose equivalent. Okay? And the transketolase is going to transfer 2. And so I take this one and I put it over here to make the fructose 6-phosphate and glyceraldehyde 3-phosphate, which are both intermediates of both the glycolysis pathway or the gluconeogenesis pathway, depending upon what I need. So the overall reaction specifically is this. 2 xyluloses and 1 ribose will make 1 glyceraldehyde 3-phosphate plus 2 fructose 6-phosphates. Unfortunately, most books will not write that. Most books will write this. 3 ribose equivalents will produce the glyceraldehyde 3-phosphate and the 2 fructose, because the ribose 5-phosphates can be interconverted. Okay? And so don't get confused if you ever see that on in another textbook or on an exam, standardized or not. What really reacts are 2 xyluloses and 1 ribose, but it could be titled 3 riboses. All right. Now, the fates are interconnected. Okay? Depends upon what the body needs. Since I am integrating the pentose phosphate pathway this way, so I've got the pentose phosphate pathway, let's glucose. The pentose phosphate pathway, NAD, well, now I can't, it's too small. Okay, NADPH is made, and ribose 5-phosphate is made, right? But we just learned that if I do the non-oxidative reactions, I'm going to be able to get glyceraldehyde 3-phosphate and fructose 6-phosphate. And I know that if I used gluconeogenesis, I can go back up, or I could use glycolysis and go down to make ATP. By using the citric acid cycle. And so there's a lot of things that could happen here. The one thing about the non-oxidative pathway is that all of those transketolase, transaldolase reactions are reversible. All right. So if that is true, there's a whole bunch of things that could happen. All right. You can get to a point where I only need this ribose 5-phosphate, but I don't want any of that. Okay? If I need to make the ribose but no NADPH, okay, can I go this way? Through those reactions that I just showed you? No, because that will make NADPH. The reason why I might not need NADPH is because I have no NADP+. That would limit the reaction. Could I make ribose 5-phosphate if I can't make this? Yeah, what would you do? You would take some of the glucose and you would make some fructose 6-phosphate and you would make some glyceraldehyde 3-phosphate and you'd use 2 of those and 1 of those, and you would make 3 of those. So under the body's condition, I could make just this alone. All right. Fate number 2, do I need both ribose and this? I just go here. That is the traditional method of the pentose phosphate pathway. What if I want a whole lot of NADPH, but not a lot of ribose 5-phosphate? I can do this. I could make some NADPH. I could take the ribose, put them back into gluconeogenesis. I could run them back up to glycolysis, to glucose, and then go again. And if I do this, I create lots and lots of NADPH without making a lot of ribose. What if I want this and ATP? I run it this way. I take the ribose and I go down and make ATP. And I can do this, run it down, and it will run any of these ways depending upon what the body needs. And so mode 2, which is I need both of them, I just do the reactions I just showed you. And I get lots of ribose, I get lots of NADPH. If I just need a lot of ribose but no NADPH, I use glycolysis and the transketolase transaldolase reactions, and I make ribose. If I need more NADPH than ribose, I run the pentose phosphate pathway, the oxidative portion, which is this part. I run the non-oxidative portions, which is this part. I go through gluconeogenesis, and I keep running this way, and this concentrations go up and up and up, and this one doesn't. The riboses don't change. And in the last, if I need lots of this and lots of this, come over, make ribose, go down to glycolysis, and continue on to pyruvate. It all depends upon what the body needs and how it will run these different pathways. So you're interacting between the 4 pathways: the oxidative pathway of the pentose phosphate pathway, the non-oxidative pathway, that transketolase, transaldolase, glycolysis, and gluconeogenesis. They all work together. Okay. How in the world do we transfer these small molecules? Okay. We're going to use enzymes that use cofactors to help them. Okay. And the cofactor that we're going to use for the transketolase is called TPP, thiamine pyrophosphate. Is there any word in that that you recognize? Thiamine. Thiamine is a what kind of vitamin? A B vitamin. Okay. And so if you do not have your B vitamins, you can't do this reaction. Now the thiamine is, a derivative is made of it. And that's the whole thiamine pyrophosphate. It's got a lot of stuff on it. But really, this is the important part right there. Okay. And so that's all I write. And you can see a base will deprotonate this. Okay. And I'll have a negative charge. That negative charge is stabilized because it has a positive charge. The electrons will attack the ketone of the 2 carbons it wants to transfer, 1, 2. And then I always draw the electrons up, but remember, it can be up on the oxygen temporarily and then attack. Now this looks weird, okay, because I have inside the enzyme, the cofactor attached to the 1, 2 carbons I want to transfer. I've got to break this bond right here. All right, I will do that by having a hydroxide in this position. Deprotonate, creating the ketone. The leaving group is this, a carbanion, which we said was really, really bad. This is where the cofactor becomes important, because this negative charge can create a resonance structure. So the electron pair really sits on this nitrogen and creates a neutral intermediate. This is really stable. This is a much lower concentration of the distribution, but it's actually the reactive species. So if I didn't have this attached to it, that bond would never break. Okay? It's because I can draw this resonance structure because of the enzyme and the cofactor that I can. Okay? Now, I said this is a very low concentration intermediate, but it's the reactive one, so this negative charge will attack on another sugar and attack at a ketone. And now I've taken the 1, 2 carbon and I'm attaching it to a new carbon. Then all I have to do is get rid of this. I do that by deprotonating, creating a ketone, regenerating the cofactor. This is how transketolase stabilizes the transfer of 2 carbons. Okay? Transaldolase. Does this sound like an enzyme that you've studied before? Called what? Aldolase. And do you remember what aldolase mechanism one was? I actually took you to a whole new page. And the enzyme became part of the reaction, and the enzyme was a lysine. Okay? Well, check this out. The enzyme and the mechanism is exactly the same as aldolase. There's the enzyme. The lysine will be deprotonated, will attack a ketone, and will create this, a nitrogen double bonded to an oxygen. Sorry, a nitrogen double bonded to a carbon where there was once a carbon double bonded to an oxygen. This functional group is called a Schiff's base. It's a very important, fundamental functional group within biological systems. Now remember, we added the enzyme on and covalently linked so that I can break a bond. I have to make a good intermediate for a bond to break. I do that by having this hydroxide in this position deprotonate, create a ketone, and this bond breaks and puts a negative charge onto a carbon. We said that was bad. But because of the placement of the Schiff's base, I form a resonance structure. This is the stabilization of why it reacts. It is mostly in this resonance form because it's totally neutral. But again, this is the reactive one. This reacts with another sugar, another ketone that it can react with, to transfer the 1, 2, 3 carbons, creating a new bond. The last thing is I got to get rid of the enzyme, so I took off water. I'll now put water back through. And if I deprotonate the water, creating a ketone back, the enzyme leaves. I've transferred the 1, 2, 3 carbons to now another R group. That is the full mechanism of the transaldolase. What's important here, okay, in both of these cases, is that to break that bond, we need to be able to create a resonance structure of the leaving group. The TPP created that for the transketolase, and the Schiff's base formed that for the transaldolase. I will not make you reproduce these mechanisms, okay, but you need to understand how the leaving group was stabilized for both of them. It is because I form the leaving group would be a carbanion, but because of the structures of how it happened, you create a resonance that is neutral. All right. If you look at the stoichiometry right, okay, if you take 1, 2, 3, 4, 5, 6 ribose equivalents, 6, see, that sure looks like a G. 6 ribose, 5 phosphate equivalents. I can produce 1, 2, 3, 4, 5 sugars back, 5 glucoses, 5 glucose, 6 phosphates. This represents 30 carbons, and this represents 30 carbons. All right? And so full stoichiometry can be had if you have multiples of 3. If you have 3, everything works out wonderfully. If you have 6, everything works out wonderfully. Okay? And so the multiples of 3 work really, really well. That's why I'm going to give you a quiz that doesn't have a multiple of 3. Okay? All right. I'm going to stop the lecture about on this particular chapter. And I want to bring up the last chapter real quick for questions on kinds of sugars that we have and how we put sugars connected to stuff. So, questions? Carbohydrates are important. One thing about carbohydrates. When we think about sugars, most of the time we really think about metabolism and we think about sugars as energy. Yes, that is important, and we do focus on it quite a bit, but sugars are much, much more diverse in biological systems than just energy. They are structural in component. They are a base by which we have functional groups. They can be informational. And so there is a lot of different ways that sugars are used in the body. And so I don't want you just to think that we break them down all the time. And so that's what we introduced in this particular chapter. They can be polymers themselves, and polymers can be storage of energy. Polymers can also be the structural components. Okay? What is the difference between starch and cellulose? Starch and cellulose. Yeah, it's just the connectivity. They're all, starch and cellulose are all polymers of glucose. Starch, in its most simple form, is just one long chain of glucoses. Cellulose is just one long chain of glucoses. The difference is one is alpha 1,4 and one is beta 1,4. Which one is the alpha and which one is the beta? One is structural and one is kind of info. The beta creates a structure in which, because everything is in chair form, I get an extra hydroxide oxygen hydrogen bond, where at the top you get covalence and at the bottom you get hydrogen bonds. It becomes structural. It's very strong. Okay? But why in the world is one nutritious and one roughage? Why can't we break it down? We don't have the enzyme. And you can test that theory by eating corn. Okay? On the outside of the husk, it's all cellulose. If you don't chew it up in your mouth with your teeth, you will get no nutritional value because it comes out exactly the way it goes in. Okay? But if you break that, you actually can extract the starch on the inside and get nutrition from it, although the outside husk comes right through. Okay? Now that's important too, because you do need roughage. You need stuff to push through, or you get all backed up. Okay? So you get some benefit from that, but I mean, that you know, we will not break down cellulose. Okay? Why is chitin even more strong? Well, it's not just sugar anymore, is it? It's not just the basic glucose. It's got derivatives. Okay? It's got some nitrogens on there. It's got some more ketones on there. It just has more hydrogen bonds. And so it can be more structural. Anything else? Yes? Could you explain the sterics of the lysozyme mechanism again? Okay, the lysozyme. Yep. Ah, okay, so the lysozyme. Yeah, that's great. Okay, so, all right, here's the mechanism itself. And it looks kind of weird. Why? Oh, stop. Done. All right. So, you have the lysozyme, and we know experimentally that lysozyme's active site holds 6 sugars, and it's going to break between the D and the E sugar. Okay? And what happens is the bond between the D and the E just breaks. Now, inside the enzyme, there is a glutamate that has a proton that can donate to make that hydroxide, right? And there's an aspartic acid that's negatively charged, so when this breaks, I make a carbocation. So I've got a negative charge next to a carbocation, and this glutamate now becomes negative charge, so I'm stabilizing the positive negative. All right, but why in the world do I break that? Obviously, lysozyme has to break that because that's what it does, and I know I stabilize it, but why in the world? That doesn't seem like it should happen. Okay? And it is because of this. When lysozyme binds the 6 sugars, okay, 5 of them fit perfectly in, okay, but the D sugar doesn't. It actually doesn't fit in the chair conformation. What happens is that chair conformation is coming down, and it's supposed to be bent down, right, at one end. But what happens is, as the other things get pulled into the pocket, this one gets bumped up against something, and it gets pushed up into a half chair. Okay? Physically kind of just being pushed against the enzyme. Now this half chair, okay, this right here looks flat. Okay? It really wants to be in a tetrahedral, but it is more shaped to be flat. If the bond actually leaves, it becomes this structure, which is sp2 hybridized, or trigonal planar, or flat. If this bond actually leaves, what's left is flat, which fits best in that shape. So we're physically pushing that D sugar into the transition state because of the sterics, which causes that bond to have a reason to break. If it was perfectly tetrahedral, it wouldn't want to break. But because it's already being forced into the structure, it actually accommodates that carbocation better physically in the shape of the active site. Okay, and that's why, that's one of the stabilizations. Now, it does help to have the negative charges next to it to stabilize the positive to the negative, but this is a underlying reason why we get that carbocation formation, because it just relieves the strain of it pushing against the enzyme itself. Okay. Why in the world does the bacteria bind to penicillin rather than to the natural substrate, which is two D-alanines put together to make the integrity of the peptide bond in the cell wall? Because lysozyme is the sugar part of the cell wall that goes in one direction, and then penicillin works against the proteins that make stability in the other direction. Why in the world does penicillin get in the way? The enzyme that binds to put together the proteins to make the structure in the other direction looks for a three-dimensional shape. It looks for two D-alanines, but it's really not looking, it can't distinguish that it's two D-alanines. It just looks for a three-dimensional shape. The two D-alanines, that shape is exactly the same shape as part of the penicillin. And so the enzyme finds the shape, and it will do its reaction. But when it does it to the penicillin, it reacts, and it's a suicide inhibitor because it can never release the penicillin. That enzyme is dead. You got to go all the way back to the DNA, upregulate a new messenger RNA. You've got to make a whole new protein to make that linkage. And by that time, your cell might have died before it can do that. And so it's just looking at a three-dimensional shape. And so the penicillin can get in the way. It would be kind of modeled after a competitive because it binds at the same active site. But once it reacts, it's a special classification called suicide inhibitor.
So the reason why penicillin stops bacteria is because it can't finish building the cell wall?
Can't finish building the cell wall, which leaves the bacteria vulnerable. Because bacteria are quite often exposed to extreme conditions, and it needs the cell wall to keep integrity. But if it doesn't have that integrity, they break open and they die away.
So even if it's off the cell membrane, it's still fine?
Mm-hmm. All right. Yes. Slide 32. I cannot remember what's on slide 32, so let's look. Yes, there we go. You don't understand? Ah, okay. So, trinag, okay, is a substrate. Okay, there's 6 active sites. All right. Now, if you go back a few slides, how much activity or how much breakage did you have with 3 nags? What was the percentage of breaking? There it is. 0. So if you have 3 of them, Lysozyme doesn't break 3 at all. Okay? So here I know D is small. Okay? So if I put a sugar in there, it's not happy because it doesn't fit. Okay? So there are 3 different ways I can fit in a trinag. Okay? I could fit them this way, those 3 places. I could also fit them in that way. Those are 3 places. Or I could fit them in these 3 places. Okay? There's only one of those that binds with 3 positive interactions. Is it the pink, the red, or the green? The green. Statistically, 99% of the time, the trinag is going to bind into the ABC slot because each one of them will give stabilization. Each one of those sugars will be stabilized when it binds into the A, B, and C slot. If it binds into the B, oh wait, there's one more. I could do one more. Sorry, you could also do this. Any one of those other ones, you're going to have 2 positive interactions that the sugars really fit and one doesn't. The only one that has all 3 fit is A, B, and C. Now, if it only fits in A, B, and C, statistically, most likely, and it has 0 cutting, then that means that can't be the place that it cuts. Okay? Because with 3, we saw absolutely no activity. And so that eliminated that possibility. Because we said we bind 6 and we break in 4 and 2. It could have broke between B and C, or it could have broken between D and E. And this kind of gives us support that it's probably D and E. And you only start getting activity, and it's not a lot, when you get 4, right? Well, when you get 4, then the only ways that you can bind are 1, 2, 3, 4, or you can bind 1, 2, 3, 4, or you can bind 1, 2, 3, 4. There's no way for it to bind without it being in one of those bad spots, right? And so that's why we get a little bit of activity. Okay, and then the more we get, and finally we get 5 positive good interactions and 1 bad interaction. That's as good as we can get. There's no more binding spots. Good. Okay, let's complicate this up. I'm going to show you and start your quiz. Okay? Now, if I'm starting your quiz, that might tell you something about this quiz. Okay? There's a reference sheet that you can use, and it shows you the, up at the top, glucose to ribulose, and ribulose down into the riboses, and then the transketolase, transalkylase, and then gluconeogenesis. Okay, so that is helpful, but you guys don't know anything about the the pentose phosphate pathway yet, because you just saw me do it. Now you're going to do it. Okay. This is the question, or this is the what you're going to do. Beginning with and only using 4 molecules of glucose 6-phosphate. That represents 24 carbons. Okay? Do not make any more carbons for me. Okay? 24. Determine the maximum number of NADPH molecules that can be produced using the pentose phosphate pathway, glycolysis, and gluconeogenesis. This will require you to proceed through multiple rounds of the pentose phosphate pathway. In the end, provide the outcomes of all the carbons, where they all are, and the number of total NADPHs. Okay? All right. So, You got 24. Now, if it was just 3, this would work out really, really well. Okay? 4 isn't the stoichiometry that we have learned. Okay? But this is what the body really does. You don't eat just 3 things. Okay? It's going to use the pathway as far as it can go, and then it will run out of something. Okay? All right. Let's try this. We have, oh shoot, I can't write on that. How do I write on this? It's defeated me. Go old school. All right. So, we're going to try this. That always gets in my way. It bothers the mess out of me. Okay. We have 4 glucose 6-phosphates at the beginning. And I'm going to go all the way through the pentose phosphate pathway. And if I do that, each one of them will produce 2 NADPHs. So how many NADPHs am I going to make with 4? 8. Okay, 8 NADPH. Okay, I got 8. That's great. Plus, okay, how many ribose 5-phosphates will I make? 4. 4 ribose 5-phosphates, plus I make something else. See, if you knew all the pentose phosphate pathway now, you know it. Okay, wait. There are 24 carbons here, right? No carbons there, right? How many carbons here? 20. There's 5 each times 4 is 20 carbons. Is that equal? What else did I produce? CO2s. So I produced 4 CO2s. That is 4 carbons to make 24 on both sides. I haven't lost anything yet. Okay. All right. Now what do you want to do? Well, you can do this. You did know a stoichiometry. You said if I took, let's say, 3 of the ribose 5-phosphates, okay, I can make some things through the non-oxidative pathway, right? What do I make at the end of the non-oxidative pathway? Well, think about that. I'm going to leave what I have left. I still have this that I have got to drop down. I just took 3 of them, so I have 1 of these left over. And I still have 4 CO2s, and I'm going to make some other stuff. Okay? Did anybody think about what is the end of the non-oxidative pathway? You get glyceraldehyde 3-phosphate, so 1 glyceraldehyde 3-phosphate plus 2 what? Fructose 6-phosphates. Okay? So here I have 3 carbons, right? Here I have how many? How many carbons? 4 carbons. Here I have how many carbons? 6-phosphate. Ooh, Apple TV. Okay. And I have this, plus I have 1 ribose 5-phosphate, plus I have 4 CO2s. Now I'm using it. And now I'm going to make something. What am I going to make? What can fructose be converted into? Fructose 6-phosphate can be converted into glucose 6-phosphate. Oh, good. I can get 2 glucose 6-phosphates. Now what can I do with 2 glucose 6-phosphates? I can go through the oxidative pathway. Each glucose will make how many NADPHs? 2, right? So I can make 4 more. 4 plus 8 is 12. NADPH. NADPH. Okay? And these get converted into glucoses, get converted into ribose. So I get 2 more riboses plus this ribose, I get 3 ribose 5 phosphates. Plus, when I change these 2 into ribose, I make 2 more CO2s. 6 CO2s. Plus, I still have this thing right here. Okay, so I took that one out and did something with it. I took this one out and did something with it. And then I took 3 of those and did something with it. Okay, let's make sure I'm okay. Okay, over here I should have 24. Okay, I have 15 carbons here. I have 6 carbons there, and I have 3 carbons there. That totals what? 24. Do you see something? Three, which is one of those perfect things. Okay? Continue. It goes on. Okay? And make sure you can react, because you might have left something over here and then you produce something and, oh, I can do one more reaction. And then I get one more thing. Okay, the first time I did this, I asked the students to do 5. It took single-spaced pages, 3 pages both sides. 4 doesn't go that long, but it still is long. Okay? Create as many NADPHs. That means you've got to go back to glucose as often as you can. You will eventually get to a place where you'll get a molecule and there will be no reactions that could happen with them anymore. But it's much further than you think it might be. Okay? Try it on your own, then get in a group and see okay, so if you have a group of 4, the 5 different ways that you came up with it. Okay? And then you are going to have to justify which one is right. Okay? This makes you think through the process. Okay? Take a picture of this. See if you can get back to this point, right, before you go on. Okay? See if you can get to here. Because to right here, you're okay. If you can't even get here, come see me. Okay?